We've been talking about proteoglycans, and I feel I might have given you an impression that all proteoglycans are secreted. That's not correct. There are certain proteoglycans which are actually integral part of the plasma membrane. Either their core protein has a hydrophobic region which is embedded in the plasma membrane, or these proteoglycans are attached to lipid bilayer by an anchor, for example, glycosyl phosphatidyl inositol anchor. This attaches the proteoglycan to the plasma membrane. Some of these plasma membrane proteoglycans act as co-receptors that collaborate with conventional cell surface receptors. Or actually, in many cases, cell surface receptors can themselves be GAGs, and they can have these GAG chains attached to them, making them proteoglycans themselves. In binding the cells to ECM, these proteoglycans, which are part of the plasma membrane, they help in binding cells to the ECM and initiating response of the cell to some of the extracellular signaling proteins, just like some conventional receptors do. So that is one of the major roles of the GAG proteoglycans present in the plasma membrane, that they can either attach the cell to the ECM or they can act as co-receptors. The extracellular domain of these proteoglycans can therefore do these two important things. Now let's talk about a specific type of proteoglycan that is an integral part of the plasma membrane, the syndecan. The extracellular domain of this proteoglycan, the syndecan, carries up to 3 chondroitin sulfate and heparan sulfate GAG chains. Their intracellular domains interact with the actin cytoskeleton and the cortex. Remember, there are many adhesion molecules we have talked about, for example, cadherins and integrins. Their cytoplasmic domain is also interacting with actin cytoskeleton. So the theme is sort of clear. So if a molecule is acting as an anchor, attaching cell to either an adjacent cell or the extracellular matrix, it pretty much has to interact with one of the cytoskeletal elements, either actin or we have talked about intermediate filaments. Fibroblast syndecans are also found in focal adhesions. They can modulate the integrin function by interacting with fibronectin and cytoskeleton and the signaling proteins. We have not talked about fibronectin. It is also a component of extracellular matrix. It helps, this molecule is recognized by several cellular proteins which are present on the plasma membrane and help cells attach to the ECM by binding fibronectin. Syndecan 4 also plays a critical role in formation of focal adhesions. Syndecan 4 cytoplasmic domain directly binds and activates PKC alpha. We have talked about PKC in one of our previous modules in the earlier course. So I hope you remember it from then. Syndecans also bind FGF and present them to FGF receptor proteins on the same cell. So they basically, you can think of these syndecans as scaffold which is going to help the cells receive the signal. They're going to capture this ligand, the signaling molecule, and present it to the receptors present on the plasma membrane. Beta-glycan binds TGF-beta and presents it to the TGF-beta receptor. It also acts as a reservoir of ligands for TGF-beta receptors. So you can store some of these signaling molecules by using these specific proteoglycans. In the case we are talking about as syndecans. Severe developmental defects can occur if a specific proteoglycan co-receptor is inactivated. We have talked about a lot of molecules. Let's look at them in a form of a grid so we get a good idea what we have talked about so far. We have talked about aggregans. These molecules have about approximately 130 GAG chains attached to it. Approximate molecular weight of their core protein is 210,000. The types of GAGs attached to this molecule are chondroitin sulfate and keratin sulfate. They are abundant in cartilage, and their primary function is mechanical support, and they form large aggregates with hyaluronic acid that we have already talked about. Beta glycan, approximate molecular weight of the core protein, 36,000. Types of GAGs, chondroitin sulfate and dermatan sulfate. Number of GAG chains is 1. Remember, we initially said that in order to be a proteoglycan, the molecule has to have at least one GAG chain attached to a core protein. So this molecule qualifies for being a GAG. They're present on the cell surface, and we have already talked about this. This binds TGF-beta and it presents it to the receptor and also functions as the reservoir of TGF-beta. Decurin, we said, is called decurin because it decorates we call decurin because it binds certain type of collagens. To be more specific, type 1 collagen. Approximate molecular weight of the core protein, 40,000, and the GAGs, chondroitin sulfate, dermatan sulfate. The number of GAG chains is 1. Perlecan, we will talk about perlecan more. Just keep this molecule in mind. We will talk about perlecan when we talk about the basal membrane. We will talk more about perlecan when we talk about the basement membrane. We have briefly talked about this previously. It is a fairly large molecule. Please keep this in mind. It is 600,000. That is the molecular weight of this protein. And the type of GAG attached to this molecule are heparin sulfate. And the number of GAG molecules is 2 to 15. As I mentioned, it is in basal lamina. The other word for basal lamina is basement membrane. Structure is one of the functions it serves as structural, of course, and also filtering function, especially which is more relevant in kidney, for example. Syndicans, we have just talked about. They have a molecular weight of their core protein, 32,000, and they have 1 to 3 GAG chains present in epithelial cells. They have a role in adhesion, cell adhesion, and they can also bind FGF molecule, which is a ligand, and present it to the respective receptor. Also, we will see that we probably won't get to talk too much about this protein, this proteoglycan, referred here as DALI, which is present in Drosophila. It is important protein because if you remember, we talked about Wnt signaling. It acts as a co-receptor for wingless signaling protein. So I hope we remember that. So we have seen that in this module that proteoglycans not only play an important role that we have thought about, we have discussed as a structure which resists compressive forces, but they have a major role in signaling and also cell adhesion.